The Institute for the Study of Peak States presents the first International Psychoimmunology and Psychobiology Research Symposium, Understanding the Institute's Approach to Psychobiology with Successful Applications, presented by Grant McFetridge.
None of this at all would have happened if it wasn't for our first speaker. So Dr. Grant McFetridge is the founder of the Institute for the Study of Peak States. He founded the organisation in the late 1990s. So it's formally as an organisation has been in existence for over 20 years. He received his MS degree in electrical engineering from Stanford University, and he's worked in research and development, consulting, and taught at Cal Poly in California. He's got a doctorate in psychology, and he's written 4 textbooks in this field that we're going to be talking about over the next few days. So it is a great pleasure that I can hand over to Dr. Grant McFetridge. Thanks, Grant.
To start this off, what I wanted to do was, because there's many people who actually haven't been trained by us or we've never met, start an introduction into what we've been doing and the whole field that we've been pioneering. I guess I just want to start by saying thank you to all the people that have been playing in the sandbox with me the last 20 to 30 years. My research colleagues and my students and patients who have worked with us over the years. And as Shane said, this was founded back in the 1990s, and it's a small worldwide volunteer organization. So that means pretty much nobody's paid for this work. And pretty much nobody's ever heard of us. We're a very, very tiny fish in a very invisible pond. So what's the core mission of the Institute? Well, it was to solve the biology behind exceptional states of mental and physical health, and to develop, hopefully, techniques to acquire that. These states, we ended up calling peak states of consciousness, and that investigation led us to places that we could never have imagined ahead of time. I still sometimes wake up in the morning and go, is this real? Did we really do this? So fortunately, after this many years, I've gotten used to it. One of the reasons that we made progress in this field is because I was an engineer, electrical engineer in particular, and my work was, as a consultant, was solving impossible problems. And so when I first started this investigation, I used that same engineering mindset, and it paid off. So in engineering, what you do is you develop models, and you figure out how to do things, and then you test it, and there's a lot of iteration back and forth. If you're wondering what I mean, think Elon Musk and his SpaceX stuff. You have models, you build stuff, you try it out, it blows up, then you go back and redo it and keep cycling around as you develop these sorts of things. So we'll be talking about that some more today. And the final thing I wanted to mention about the Institute for the Study of Peak States is what I consider one of the most important things that we did as an organization, and that was our pay-for-results policies. So when we do stuff with paying clients, we want to make sure that they actually get services for their money, especially because a lot of the people we see are struggling. We want to make sure that they get value because they need their cash to try other treatments if the stuff that we're doing doesn't work. And so the people who work with me all have a pay for results policy in what we do. And as you can imagine, that's very unusual in this field of psychology and medicine. That we've published a number of textbooks in our work so that you can actually go to Amazon and buy these things and read about the stuff that we'll be talking about today. As I said, this is going to be an introductory review. I'm just going to be going through the individual, topically, the individual discoveries that we've made, and maybe tell a few stories because we've got time for it. Okay. Oh, and one of the common questions we get is, why are we unknown if we're doing such amazing work? Well, there's a couple of reasons for that. One is what we're doing is rather unbelievable. So that's the first problem. Second problem is we've been rather unsuccessful in getting published. Our work is so different from a lot of other people's that most of the organizations we approached weren't interested in having anything to do with the stuff that we're doing. So we just kept working on our own. And the final reason is actually a practical one. We started to get much more known back in 2006, and what I discovered was suddenly it didn't have any time left for research. And we had to stay focused on our project. This stuff is taking a lot of slow development to work out, and it's easy to get swamped if you have too many people. So we contracted into just keeping a small team, and that's worked out really well for us. I don't know if you can see me in the background here, but over time, what's happened for us is that we've figured out more and more stuff. And as we've done that, our ability to solve problems has been going quicker and quicker as our models got better and better. And at this point in our work, we're actually able to do stuff fairly quickly that I had only dreamed of 10 years ago. Okay, so why are the discoveries we made actually important? Well, first of all, they give us new ways to find causes for many diseases that have never been understood. So what do I mean by that? There's a lot of chronic illnesses that current biology models don't explain. People have these problems, no one knows why. They can observe the problem, but they don't really know what the reason is. And because they don't know what the reason is, treatments aren't available, or at best, the treatments are only topical. In other words, they help reduce symptoms. What's pretty exciting about our work is we can actually use these tools to figure out what's really causing people's suffering in a particular disease sense, and then either work out a psychobiology treatment, or it can be used complementary with existing biological allopathic medicine. Okay. What else is important about our work? Well, it's cheap. Both the research and the treatments are cheap. And if you know medical science as it is presently, the word cheap and medical science don't generally go together. So people can actually do this stuff for a minimal cost. And what's more, the work can be done basically in your home. Well, that's what we do. We mostly work out of our homes. So the research work can be done very easily, well, very simply and very cheaply. How does this impact things? Well, once the research has been done and you develop a process, you can implement this in the world very inexpensively. What's even more amazing is that in some of the cases, some of the treatments are so simple that you can do a YouTube video and then have people just follow along and get a decent rate of success. We tested that whole principle about 2005 with a video on how to do the inner peace state, and that's worked out pretty well. It works about 30% of the time, just by watching and following along on a video. The next thing that this stuff is important, and I wrote this actually about 6 years ago or 5 years ago. There's two issues that are going on in standard medicine. Antibiotics are becoming less effective. Superbugs are becoming more and more common, especially in hospitals. And the other problem is actually medicine itself. There's a lot of chronic illnesses that people have no clue on how to treat, and they're becoming more endemic around the world. And the third reason actually turned out to be kind of what we're experiencing now, and that's worldwide pandemics. With psychobiology techniques, people can work on these diseases that are pandemic in ways that can be done very, very quickly. Now, it doesn't work for everyone, but for the people it does work for, you know pretty much instantly whether you were successful or not, because the symptoms change. And when I wrote that on the website 4 or 5 years ago, I was thinking about the possibility of pandemics because of the Spanish flu in 1918. But now we're actually in that position, and that's one of the reasons why we're having this conference, because my colleague, Dr. Lykkegaard, will be talking about our work on coronavirus, I believe tomorrow. Okay, and the final reason I wanted to share this stuff is because one of the things that really bothered me when I first got involved in the field of medicine was how unlike engineering it was, that there was no underlying unifying force or model that tied biology, consciousness, treatment, psychology, all of those things together. And as you'll be seeing shortly, the work that we did actually allows us to tie all of those things into one very elegant model that explains how all these things work together. I kind of like this slide because the work that we're doing in psychobiology is what is often called a disruptive technology. So what's that? It means a technology that can do things that existing technologies can do, or it can do things that existing technologies cannot do, and it can usually do it simpler and faster so that it really makes a difference. So what you see with existing technologies is that they start slow. Applications are pretty much the same applications that everybody has. You're figuring out the technology, but as they become more and more mature, you figure out more and more stuff, they start to be able to do things simpler and faster and cheaper than existing things. So what's a good example of that? The iPhone would be a very good example of a disruptive technology, or digital watches or digital cameras. They basically replaced a large industry. Now, I don't see that exactly happening with what we do, although some of the stuff we do cannot be done in any other way that I know of. Psychobiology can do things that standard medical things can't. But it's one hell of a complementary approach to medicine. And over time, I could see that as the world changes, it could be incorporated into medical practice, or that's what I'd like. Okay, so what's the bad news? Well, the good news is it's a disruptive technology. The bad news is it's a disruptive technology. So this means that it's difficult to introduce to existing organizations because it requires retraining, a new mindset, and interactions with disciplines that are currently considered very, very separate from each other. So for example, you look at, say, Teslas, which are a disruptive technology in the automotive field. Most of the car companies who are very comfortable with what they're already doing simply don't want to face the need to change. They're comfortable with what they have. They have existing technology. They know that market. And so disruptive technologies are difficult to get into existing systems. Next problem with what the bad news is that we're still developing. We've got tons of basic research still to figure out. And so people come to us and say, well, I need this and this and that disease cured or this other thing done. We're just not ready. We're just a handful of people slowly working out the biology and the psychology behind these things. The good news is a lot of the things that we figured out are allowing us to do this faster and faster, faster. And as we publish these books, we can share that with other smart people who can run with what we've done. Again, we've got decades of work ahead of us to become a mature technology. Right now, it's basically a startup. So you're looking at just the first few years of whatever thing that you're interested in describing, like SpaceX, iPhones, pretty much any startup industry. Okay. The last thing is this approach won't work for everyone. Some people can't do the techniques well. That's why I consider it in some ways a complementary to standard medical practice. The final thing is it's not magic. It means that you've got to pour a lot of skull sweat into the research. And this isn't like Harry Potter, where you just wave your wand and make something happen. It actually is real science. And that means real science is complicated and takes a lot of work. And yeah, it's like that. Okay, so let's take a look at the field. And I'm hoping that there's people in the field who are actually who are online today. And I'm sure that they have their own experiences of psychobiology and all of the rest of this. But from my perspective, and again, it's just my perspective, in the 70s and 80s, there was real excitement around this whole field of psychobiology. And real fundamental discoveries were being made. People were acknowledging the existence of phenomena that had never been really recognized before. But it was pretty exciting. For example, the placebo effect people really started paying attention to that. Now, for those of you who don't know, the placebo effect is where you can take something like a sugar pill and get well if you believe that that pill is going to help you. The thing that a lot of people don't realize is the placebo effect actually makes you well. In other words, it isn't that you think you get well, you actually get well. So you could think of it as an alternative to drugs. The problem was, when people started researching this, they couldn't figure out how it worked. And that initial excitement in the 70s and 80s basically faded over time. So this area of research, people started moving away from it because they felt that they weren't succeeding. And it turned out that this was a really hard problem. And there's a reason we spent 30 years working on this stuff before we were able to start solving these issues. Okay, so as people shifted away from excitement around psychobiology, they went other directions, and one was into trauma therapies. In the late 90s, some of the trauma therapies that are in use now became available and spreading out, and alternative and complementary medicine started to make inroads into the public. Less so into the academia. They were sort of forced into it because people were starting to pay for it and take their money to try and get help with a lot of the chronic diseases that medicine couldn't handle. Okay, so that's looking back. Now, from our perspective, this is the timeline that we had. We started with trauma therapy back in the 90s. And so for about 5 or 6 years, that was my real focus. Then we moved into developmental psychobiology, and I'll describe what that is in a minute, but it has to do basically with early development, basically prenatal and earlier development. Then we moved into subcellular psychobiology starting around 2001, and we worked on that for about 10 years. And then we got into psychoimmunology around 2017. So just here's a picture of how these all fit together. So the trauma techniques are sort of core to this work, because if you don't have a good trauma technique, you can't implement these discoveries. Then developmental psychobiology has to do with early development. Subcellular psychobiology has to do with what's inside the cells, and psychoimmunology has to do with how you develop techniques that make you immune to various diseases. So let's just talk about the first breakthroughs in trauma regression therapies. There's basically we discovered that there are 3 kinds of trauma. The most standard one, or the one that people are mostly familiar with, is what we call biographical trauma. That has to do with things like post-traumatic stress disorder. It's like when you got slapped when you were a kid, and that memory is still painful. There's a number of therapies that work on that. And back in the 1990s, when I was first starting this work, there weren't really any good trauma therapies available. And so the first project that I had was to develop a trauma therapy so I could start to eliminate stuff to figure out what was going on. And I called that, for better or for worse, I called that therapy wholehearted healing. Sort of wish I'd just called it some McFetridge's regression technique or something now, but it's too late. So that technique allowed me to actually eliminate trauma. And it's from there, it did something else that was even more important. It allowed me to examine trauma in prenatal events. Anyway, back to the other kind of trauma. There's another form called generational trauma, and that type of trauma is inherited. And it's really an important kind of trauma. It's called epigenetic damage, and that it's really important because it affects how your cells work, basically their structure. So incredibly important kind of trauma. So when you get a disease, say that's shared among your relatives, that's often generational trauma caused by epigenetic damage. And the third type of trauma we call body associations. And that type of trauma has to do with addictions. It's indirect cause for schizophrenic voices and a bunch of other issues. Basically, Pavlov's dog, the dog that you ring a bell and it would salivate, has to do with body associations. Okay, so we figured all that out, and we figured out simple ways to deal with those three types of traumas. Okay, so now we had a tool, and we used that tool to look at prenatal trauma. In particular, we wanted to look at prenatal developmental events. So these are key moments in development where the organism changes, not just moments where you got bashed, say in the womb, but instead moments where you radically changed your biochemistry. So what's an example of that? Implantation. You went from a free-floating little blastocyst into something embedded in the womb wall. Conception, huge change when you went from being an egg and sperm into something new. Developments where ovulation, where the egg is being released out of the ovaries. So each of those kinds of events have a huge impact in the biology. Now you say, well, that was a long time ago and it doesn't matter. But it turns out it matters a lot, because for most people, those events don't go well. I mean, you survived. You're sitting here in this room at this point, but you didn't survive optimally. And so as we started to study these events from an experiential viewpoint, we started to realize just what influence these key developmental stages, or I should say more like events, because they're very short in time, impacted health in the present, both mental and physical health. Okay. So I've been in this field for long enough that this seems completely ordinary, but when I talk about this stuff to people who aren't familiar with this work or this field, they're not aware that people can regress to, say, the womb or being an egg or being a sperm and experience it with emotions and feelings of injury and all sorts of, as if they were really there as an adult. But it turns out that it's the case. And there's a number of organizations over the years that have been working with this sort of stuff. It's generally called cellular memory in the field. Okay. Yeah. So anyway, now we had a tool. Now we are examining these events. So where did it go from there? What's next? Okay. Now this is very cool. Okay, up to this moment, old boring stuff. But this was exciting. So I'll just tell a little bit of a story. Over the 10 years or so in the 90s, I was doing spiritual practices and psychological techniques and all sorts of stuff. And I lived in California at the time. And so there was just a lot of stuff you could do. One of the things I did was I meditated for a week and then had acupuncture and did breath work and went into a sweat lodge, basically in a sequence there. And during my time in that sweat lodge, I suddenly acquired an experience that I'd never even seen or heard of before. So here I am, and it's hotter than hell, and I'm having troubles breathing because the air is really tight. You're sealed in this room, you're packed in with sweating people, and all of a sudden, I had a lot of room. And I had the feeling that I was on the dirt floor in a huge space, stars overhead. There was these stone monoliths kind of in the distance. It was amazing. And I went, wow, this is really strange. And then I could kind of bring myself back to the room where I was with all these people packed in with me, completely dark, smoky air. And I just felt so relaxed and with so much room. Okay, well, that was interesting, but the real interesting part is afterwards, it didn't go away. So whatever I had done seemed permanent, and I was able to induce this state into my colleagues, in particular, Dr. Adam Weissel, an MD in Israel, who was my co-partner in most of his research. And between the two of us, we started playing around with this state of consciousness. What was most noticeable about this when we first were able to teach it to other people was what looked like seaweed. So we're looking around going, this is very strange. And there seems to be an ocean floor there with a bunch of seaweed sticking out of it. And so we discovered empirically that if we dragged some of the seaweed out of the ocean floor, trauma would disappear. And we went, what the hell? And so anyway, Adam and I were working on this, trying to figure out what it was. And I think about simultaneously, we came up with the idea, my God, we're looking inside a cell. Now, I think he figured it out. He says, I figured it out, but whoever figured it out, we realized that up to that point, we had been working with subcellular biology and not even knowing it. Now, why didn't we know it? Well, a lot of people don't realize that the stuff in the textbooks, the drawings and stuff, is based on pictures from electron microscopes. And you got to understand how they make pictures like that. What they do is they take a sample, a tiny sample, they slice it into slices, and then they freeze dry it, and then they do electron microscopes on it. It's very much like trying to reconstruct a cow from a piece of beef jerky. Okay, so what we were seeing in the textbooks and what we were seeing in real life looked very different, and it took us a while to figure out what we'd actually discovered. Okay. Now, having said that, it turned out there were a lot of problems with what we discovered because we were able to interact with our own biology. And remember that seaweed we were pulling out? Well, it turned out that those were mRNA strings that were stuck into the in the nucleus, and by pulling them out, we were actually harming ourselves, although we didn't know it for about a year or so. So you can interact with your cell in ways that actually damage you. Okay. So that's the first part. Well, that would just be an academic curiosity, except it turned out that there was a huge breakthrough in there that we hadn't even realized at first. And that was, we were in a single cell in the body. So what happens is that when you have this particular state of consciousness, you pop into one particular cell in the body. Well, we ended up calling this the primary cell because it turned out that there's only one cell in the body where consciousness is. All the rest of the cells in the body are extensions of that particular cell, and that cell forms on forced cell division after conception. So why is this important? So if you think about this one cell, it's like you're that one cell, and everything else you're sort of running. It's like it's a coat you wear. Your body's like a coat from the perspective of that one cell. So our experience is of the primary cell is overlaid on our experience of the world and our body itself. So if you have a pain on your body, that's the multicell lever. But if you have some damage inside the primary cell, you'll have pain that'll feel like it's on the body, even though it's actually pain from the primary cell itself. This is an incredibly important discovery because we realized eventually that if you change the primary cell, everything else in the body changes. And that the issues that people suffer from are based on problems in the primary cell. And what we had done was we'd accidentally figured out how to interact directly with the primary cell. And over the next 10 years, we figured out techniques that allowed us to modify that cell itself. So as we started to develop tools that allowed us to interact with the cell, this one particular cell, we call this subcellular psychobiology. The psycho part because these tools looked psychological or kinesthetic, and the biology part because we were actually working with biological structures inside the body. And the next thing that we discovered was that the primary cell was loaded with pathogens in a normal person. And these pathogens were causing a lot of problems. And in fact, you could say that these pathogens were making people average. We were like, each of us is like one of those big old trees in the Amazon. They're covered by creepers and they've got things living on them and little animals and stuff like that. So we were like this huge support system for other organisms. And unfortunately, these other organisms were pathogenic. They weren't symbiotic. They were instead causing minimal or major problems inside of people. Okay. So like I said, over the next 10 years or so, we started investigating the primary cell and developing tools that could interact with it safely. Okay. And in 2014, we published our, 2006, we mentioned that in one of our textbooks that there was a primary cell. In 2014, we published a handbook on what we discovered cases of problems inside the cell and how to treat them. Well, it turns out there's something called the triune brain structure of the brain, I should say, the triune brain structure. And we know it colloquially as the mind, heart, and body. Okay. Back in the 60s and 70s and 80s, and I think he published in 1990, Dr. Paul MacLean at the National Institute of Health analyzed primate and human brains and realized that they were basically three structures in one. And he called it the triune brain. And it was really a fundamental discovery in biology. And people had realized this for a long time, the mind, heart, and body, but they didn't know the biology behind it. Well, it turned out that there was more biology behind it than that even we realized at the time. And that turned out to be inside the cell itself. Okay, so inside the eukaryotic cells, and those are cells of organisms that you can see in the world around you, there are different membrane structures. And these are a legacy of evolution, that billions of years ago, a bunch of bacteria got together and formed a single organism, and they all shared their lives basically together. So they unified to become one organism from being individual organisms. So they went from being a bacteria, bunch of different kinds of bacteria, into becoming a eukaryotic cell. That's called the endosymbiotic theory, and Margulis discovered that. And after taking a lot of crap for nearly 20 years, people recognized that she was correct about it. Okay. So it turned out that what we discovered, because we could work with the primary cell, was that these mind, heart, and body structures that we think of being in the brain were actually reflections of stuff that were happening inside the primary cell. That the primary cell was made up of these different bacterial entities from evolution, but they had retained their individual awareness inside the cell. So I'll say that again. Imagine that there were 7 bacteria that got together billions of years ago and became one organism, but they didn't give up their individual consciousness. And it turned out that that model explained a lot of things. And so it turns out that our subconscious, based on what's happening inside the cell, is a committee. and often they don't get along, and often they have competing objectives. Right? So if we look at a cell, these different structures in there correspond to different parts of our body. The heart, where you have emotions; the belly, where you may have fear; your mind, where you think. Each of these different things that we think of as in our body are actually reflections of stuff that happens inside the cell. It turned out that there's a level of smallness that goes even beyond this. Inside the nucleolus itself are these tiny, tiny little cubical structures that we call blocks that form the basis of all of these structures. So it's like Russian dolls. These tiny little blocks are where the awareness is. Then there's the subcellular structures in the primary cell, and then there's the multiple cell structures out in space. And so like Russian dolls, they just echo out and they superimpose on each other. Awareness is composed as if it's all one thing. The tiny structures of the blocks, the stuff that's happening inside the primary cell, and then the multiple cell structures. They all share awareness, a common awareness. This is an interesting idea, and some people may be going, well, that sounds like garbage, but it turns out that it explains trauma at a biological level. And that's an incredibly important thing to understand, because once you understand that trauma is actually based inside the cell, you can start to design techniques to get rid of trauma that the world's never seen before, because you actually understand what you're trying to do. So if we look at a trauma experience, say when you were slapped when you were in second grade. And when you think about that experience, it still bothers you. You can sort of feel the slap and you feel shame and all the rest of that sort of thing. It turns out that that experience is stuck in your psyche because of a problem inside the primary cell. It turns out that when you need to do things in life, you need proteins to actually do stuff. And the way that's done is that a gene is what they call expressed. So a gene inside the nucleolus comes out to the edge of the nucleus. It's copied in mRNA, which is called messenger RNA. And then it's sent out a nuclear pore out into the cytoplasm, where it's read by a ribosome. The ribosome reads that string like it were a ticker tape and creates a protein. Well, that's what happens if everything works well. What happens with trauma is that when the gene comes to the inner surface of the nucleus to be copied by the mRNA string, it's copied. The mRNA string comes out of the nucleus, but it's still stuck to the gene. So in this illustration, you see the edge of a nuclear membrane, an mRNA string coming out, and a ribosome trying to read it like a ticker tape and getting stuck. It turns out the structure directly corresponds to trauma. Now, if you remember when we first discovered this stuff, we yanked out what looked like seaweed from the ocean floor. Well, what we were doing, we were yanking out this mRNA string and essentially dissolving trauma. What we didn't realize is that we were damaging the nucleus when we did it. That inside the nucleus, underneath the bottom of the seafloor, was genes. And the genes weren't getting fixed, and instead we were just wounding the nuclear pore by doing this. It took us a while to figure this out and then come up with techniques that could easily repair the interface between the DNA and the mRNA and actually cause trauma to go away quite simply. On the second half of the picture, you see this mRNA string superimposed on the person's body, and that's actually how it works. You feel the effect of a trauma in a different in a location of your body, and the two are superimposed. The feeling of the ribosome and that's embedded in the ribosome, and the feeling in your body. Okay, so all this is interesting, and that brings us up to about 2017. So what is psychoimmunology? Well, psychoimmunology is the use of psychological or kinesthetic techniques to make a person immune to a targeted pathogen. Okay, so up to this point, we've been modifying stuff inside the cell, but still loaded with diseases, and we want to get rid of them. We don't want to just be a jungle tree that isn't bothered by its parasites. What we want to do is get rid of the parasites themselves. So we started working this in 2017, and we immediately rammed up against problems that we needed to make new discoveries to make that work. And so we've been working on that for the last 3 years or so. And in fact, we wanted to start on viral research in the fall. So I was tentatively scheduling viral research to happen in September, October timeframe. And then the coronavirus hit. And suddenly we changed our priorities and started to work on how viruses work and how to get rid of them, and started making breakthroughs, which my colleague, Dr. Likkegaard, will be talking about tomorrow. Okay, so having said that, it's a new technology and it's real. It's real science, and you can do real stuff with your cell, and you can screw up your cell. And so we spent a lot of time on safety issues. And in fact, we had an agreement in the people in the Institute not to share our research for, from 2001 to 2014 about the primary cell, because we realized that there were safety issues that we hadn't solved. So it wasn't until about 2012 that we started to feel that we'd solved the safety issues well enough to talk about it with the public. So having said that, clients, when they read our books on research, get kind of concerned because we talk about safety issues quite a bit. What they don't realize is it's just like working with drugs. In drug development, you have research, then you have testing, and then you have sales in the grocery store or in the pharmacy. And we have to basically have the same structure for the same reasons. So in research, the risk is quite high. You can screw up. You can do interventions because you basically don't know what you're doing and harm yourself. And so a lot of our work is with research scientists who agree that the results are really important and that it's worth possibly damaging ourselves because these discoveries are so important. So think someone like Madame Curie, who investigated radioactive substances and died from it. She and her colleagues felt that this was so important, and they also didn't realize all of the risks, just like in our case years ago. Okay, so that the research phase is highly risky, and we tell people don't do it unless you recognize the safety issues that you're facing. Now, having said that, again, we're further up the curve. We know a lot more. The risks are getting less and less because we understand more and more, but we're still exploring the unknown. Okay. The next step is testing. So we test out processes on healthy people to see if there's going to be any abreactions, any problems. And after that stage is done, then we start working on the people that have the problems that we were trying to help in the first place. Finally, after a couple of years of work, we release it to therapists who are aware that these are new processes. They work with clients who are also aware that these are experimental processes. And then we start seeing more and more people use these sorts of things. Eventually, we feel it's deemed safe enough and we understand the pros and cons well enough to publish. All that can take 5, 6, 7 years, depending on the process. Sometimes it goes quicker, sometimes slower, but it takes a while. Okay. The next thing I'd like to say is, okay, you've heard all this sort of thing, and how do you know it's true? It could just be the delusions of a handful of crazy people scattered around the world. And if I were you, I would also suspect that. I've been working in this field, so I've gotten used to this sort of thing. But how do you know? Well, how do you know that radio waves exist? Can you taste them? Can you touch them? Can you smell them? Nope. So how do you know that they exist? Well, is it just because everybody says they exist? Well, for some people, that's probably true. But the real reason that people know that electromagnetic waves exist is because if you understand that they exist, you build a model that they exist, and then you can implement things that have never been done before. So if you go back to the late 1800s, early 1900s, when telephone and motors were just being developed, they were working with these invisible models to create things that had never been done before. Well, the primary cell and the stuff that I've been discussing up to now is the same way. You don't have to believe it. And in fact, without personal experience, I think I'd be very surprised if you did believe it. And I'd worry some about your sanity. However, what you can do is take it as a model. And if the model is real, you can then figure out implications to the model. In other words, if this model is a good model, maybe it's not even real, but if it's a good model, you can then say, well, if I do this and this and this according to the model, this should happen, and then you can test it. And that's exactly what's been going on in our work. Now, I have to say that because we can see inside the primary cell, that makes the model a lot more vivid to us, but that's not necessary. You can just assume that it's just a construct that's very useful, and then go from there. And it's been very useful. Okay, so having said that, we've released a number of techniques. These theories don't help unless you have ways to intervene or interact. We've published different things in various ones of our books. We've also published a lot of theory books, and you can go and get these from Amazon and read about them and the pros and cons and how well they work or how well they don't work and that sort of stuff. Applications. There'll be a lot of talk about applications over the next 3 days, but some of the applications are how to make trauma heal much more quickly and simply. When you realize that you're working with trauma that's actually due to structures in the cell, you can target the cellular structures in ways that people never imagined was possible. You can eliminate a huge variety of psychological and physical symptoms in clients. Now, the reason we have so much emphasis on that is because we were looking at the psychological side, what was causing that, and then figuring out the biology. And so we have an entire handbook written for therapists, basically on mental and some physical disorders. And that's where our focus was for a lot of years as we tried to work out these models. Schizophrenic voices, we were able to finally figure out what caused that and simple ways to eliminate it. It turned out that we also found something kind of shocking. We found that there's a common disease in most people that causes multicultural conflicts. It's a fungal parasite, and once you make yourself immune to that, boom, people just feel like people. There's no division anymore between cultures. We found what causes Asperger's syndrome and have a very simple way to get rid of it. We found what causes OCD and a way to treat that or get rid of that problem. Turned out it's a fungal issue, and one of my colleagues will be presenting that, I think, today. We found out a simple way to get rid of the symptoms of traumatic brain injury for many people. And Dr. Pellissier, our MD colleague in the United States, will be discussing that, I believe, tomorrow. And I'll finish up, I guess, tomorrow at the end of the session with some of the disease treatments that we have in our research pipeline. That's pretty interesting to me. Yeah, and finally, I'd like to say the reason we did this conference was to invite other excited, capable researchers around the world to play in the sandbox with us. There's a lot of things that people know that we don't know. There's stuff we know that other people don't know. A combination of that will change the world. And that's why we're doing this. That's why we volunteer our time, have our very limited time to do this sort of work, risk our lives in some cases. Yeah, it's because we want to change the world and we want to move this out into the world. So if you're listening to this talk and you got a little inspired, it's very early in the morning for me, so I'm not quite as jumping around as I normally do when I talk. Please get in touch with us. Grant@peakstates.com is my email address, or you can phone me at home at 250-509-0514. Okay.
Thank you for watching. For more information, please visit peakstates.com.